just like we have excitation contraction coupling for skeletal muscle, we do for cardiac muscle as well. All right, so let's orient you. Here's the sarcolemma, that's the muscle cell membrane. This is cardiac muscle cell, okay? Cardiac muscle cell. We still have T-tubules. They are not as fancy, they're not as well defined as they are in skeletal muscle, but they do exist and they do work, all right? So here is our action potential traveling down the cell membrane into the T-tubules. Now here, instead of DHP receptor, we have a different receptor, calcium channel, voltage-gated calcium channel. When this action potential reaches here, it causes that calcium channel to open. Calcium comes rushing in, okay? The calcium increasing, the increase in intracellular calcium concentration then causes additional calcium to be released from the sarcoplasmic reticulum. All right, so it's a two-step process. Calcium comes in from the outside. That calcium then triggers the release of additional calcium from the SR. If calcium doesn't come in from the outside, there's no calcium release from the SR. Do you see the difference? Very different than skeletal muscle where it all comes from the SR. Okay. Here it's coming from the extracellular fluid. This actually points out an important clinical point, which is if your calcium is too low, your heart won't contract. Believe it or not. Why? Because there's no calcium to enter from the outside. So there's no SR calcium release, so there's no contraction. All right. So we still, so calcium enters, calcium, the sarcomere function is very similar to what we see in skeletal muscle, right? Calcium binds to troponin, troponin moves tropomyosin away from the binding sites on actin, myosin and actin do their cross-bridge cycle thing. The difference is that the troponin and tropomyosin are slightly different molecules, right? So they're not the same protein, but they do the same thing. Okay, so the calcium in the cytoplasm, these are the ryanodine receptors in the SR membrane, so that's different than the DHP receptor. Just pointing out that it's different. Okay, now when it comes to relaxation, all right, so the way relaxation occurs is when calcium concentration in the intracellular fluid falls. How does it fall? Some of it is pumped back into the SR, okay? Other of the calcium is pumped out using a sodium calcium exchanger that is in a sense powered by the sodium potassium pump. So when these two things have happened enough, then the heart muscle contracts. Troponin moves back, troponin moves tropomyosin back over the covering sites. Myosin can't bind to actin, so we get relaxation. Now, the interesting thing in the heart is relaxation never occurs for very long, right? Remember, everything's a cycle. So by the time the calcium is pumped out and the heart relaxes, by the time it's good and relaxed, then the whole thing starts over again, right? So it is kind of a pattern.